Okay, everyone, welcome back to Chapter 24. We're halfway through the material for the second exam, so let's crack straight into it. So, Chapter 24 starts on the chromosomal basis of inheritance, which basically picks up where Mendel left off, because now we actually know what these heritable factors that he saw in his work were, and that is the actual DNA, and we know that there are traits linked on the individual chromosomes. And they have, each chromosome has multiple alleles, and they're in a particular order, and something that you didn't really see in Mendel's work, because he just so happened to pick traits that were not linked, is we have linkage groups. And those are alleles that are on a single chromosome that are inherited together. It just so happens that the ones he selected were on separate chromosomes and completely unaffected, and he just kind of assumed that's how all of it worked, but we know better now. And if something is referred to as sex-linked, then it is on one of the sex chromosomes. Almost all sex-linked traits are on the X chromosome because it is simply much larger than the Y chromosome. In fact, most of what's on the Y chromosome is a little bit redundant, I guess you'd say, because everything you need to make someone either male or female is contained in the other chromosomes, for the most part. You just need a couple of genes in the Y chromosome to turn on the machinery to switch over from the genetic default of female to male, which is part of the reason why male fetuses are a little less hardy than females, because there's a lot of switching over that has to be done. So anyway, that aside, alleles that are not linked show independent assortment. Those that are tend to be inherited as a group, and the frequency of cross-link or crossing over can tell you the sequence of the alleles on a chromosome, simply because the further apart two traits are, the more often you'll see them inherited separately, and the closer they are together, the more likely they are to be inherited together. Just because if let's say you've got your G and Z up here, and you randomly insert a crossover event, you've got this whole space in between them here where that can happen. High probability. But if you're talking like S and R, well, the majority of the area here is on either side of it, so you have to have something land right in between it to where it crosses over, so that's not terribly likely to happen. So normally, we've talked about this a little bit before, but normally you have 23 pairs of chromosomes, 22 are autosomal, and one pair is your sex chromosome. For females, you have two X chromosomes. For males, you have your X and Y. And just to avoid any confusion, we are talking about biological sex here. We're not getting into any social studies or anything like that. The genes that are on your sex chromosomes are referred to as control sex-linked traits. X-linked are on the X chromosome, and that's the majority of them because the X chromosome is just so much bigger than the Y chromosome, and there are no matching genes for it on the Y chromosome, and the Y-linked traits are found on the Y chromosome, and again, there's not a lot there. You can see right here in the diagram, the X chromosome is almost the same size as, or bigger than, the majority of your somatic chromosomes, and the Y is the tiniest. Because again, everything you need to make a male is on these other chromosomes. You just need a couple of genes to activate that circuitry, basically. So X-linked recessive inheritance, or I'm sorry, sex-linked inheritance. Most X-linked traits are recessive, therefore you're going to see them a lot in males because males are hemizygous for the X chromosome, meaning they only have the one. And for females, they would have to inherit two recessive copies of a X-linked trait, so you're not going to see them terribly often in females. Most of the time, they're going to be a carrier. Color blindness is the classic example of this. Very few women are colorblind because they can have another X chromosome with the dominant copy of the gene that allows you to see red, green, or the difference between red and green. And so then they're just carriers of the trait. A father can only pass on his Y chromosome to his sons, so you cannot get color blindness from father to son. It has to come from, for a male to have it, it has to have come from the mother, because she can only pass on an X chromosome, so there's a 50-50 chance of her passing on color blindness to her son. The only way to get a colorblind female is if you have a mother who is either colorblind or a carrier and a father who is colorblind, because the father can then only pass on that one colorblind X chromosome to his daughter, and then depending on whether the mother is hetero or homozygous, she either has to or has a 50-50 chance of passing on the colorblindness trait to her daughter. You'll typically see this, and see if we can zoom in without breaking the computer. You'll typically see something like this, where it follows kind of a recessive pattern in that it skips generations, but the majority of the time that you will see the trait show up is going to be in males in the family. It very rarely will a female display the trait, but you can see here the grandfather was affected. He had to have passed on this affected X chromosome to the son here. I'm sorry, he passed on the affected X to his daughter and an unaffected Y to the son. This over here is another colorblind female who's bringing the trait from a different gene pool she married in. And you can see a couple of generations down the line here that the colorblindness shows up again, because the grandfather gave the daughter the colorblind X chromosome, and she married someone who was not colorblind, but she has a 50-50 chance of passing it on. So one grandson is affected, one grandson's unaffected. So things like that is how you will be able to recognize a sex-linked pattern. Other X-linked disorders, you have Duchenne's muscular dystrophy, characteristic wasting away of muscles, and they tend to be kind of shriveled up. The joints locked because the muscles just kind of contract and shrivel and waste away. Fragile X syndrome, most common cause of inherited mental impairment and most common cause of autism. And hemophilia, there are multiple forms of hemophilia, but they involve the absence or minimal production of clotting factors. And if you're missing out on a clotting factor, which there's this whole cascade that has to take place in order for your blood to be able to clot correctly. If you're missing any one of those, or you don't have enough, you cannot clot your blood. But kind of like insulin, you can give someone clotting factor injections so that the way they function normally. So it is a relatively treatable condition. And this is just a pedigree of the royal family and tracing hemophilia through their bloodline. The problem with the royal families of Europe is they tended to intermarry with each other. Like, you would only marry another royal, whether they be, another royal from the same country or from a different one. And eventually what happened is they were all basically inbred to one degree or another. And a consequence of that is you tend to see a very high prevalence of these recessive traits within populations that have a limited gene pool. So hemophilia, at one time, was known as the royal disease, because that's really about the only population it seemed to affect. Practice problem here, it's got the answer right down here, but what you need to do is try to go through and figure out figure out the genotype and phenotype of the offspring from this pairing in the practice problem. And if you can do that, you'll be in pretty good shape for the exam. So changes in chromosome number. Normal development depends on exactly 2 of each chromosome. If you have a trisomy, which tends to be better tolerated than a monosomy, you can survive with some of them. Down syndrome is trisomy 21. Reasonable chance of survival, and the reason that tends to be better tolerated among the trisomies is because the 21st chromosome is pretty small. And the other thing is if it is a trisomy or monosomy of the sex chromosomes, you have a pretty decent chance of surviving that too. For most of the others, you have virtually no chance of survival, especially for a monosomy. This can occur through non-disjunction. So during meiosis 1, which we talked about previously, when both members of a homologous pair go into the same daughter cell, or during meiosis 2, when the sister chromatids fail to separate and both daughter chromosomes go into the same gamete. The result is either a trisomy or a monosomy. Monosomy, you get 1 copy. Trisomy, you get 3. Fairly self-explanatory. And that's diagrammed here on this slide if you want to take a look at that in more detail later. If you have abnormal numbers of sex chromosomes, it's usually not that big of a deal for, depending on, like if you have extras, it's usually not that big a deal. If you have too few, that's where you can get a problem. So if you have 1 X chromosome, you have Turner syndrome, and we'll talk a little bit more about that further down if memory serves, but there's some slight mental impairment, kind of a webbed neck, short stature if I'm remembering correctly, and they are an individual with Turner syndrome are infertile. But you do get extra bar bodies because they're just inactivating with the Turner syndrome, you don't have bar bodies. For poly X and XXY, poly X females and XXY males, you get extra bar bodies when you're inactivating the additional X chromosomes. So it tends to function fairly okay. And you can have multiple X chromosomes, but a single Y will always make you male no matter how many X chromosomes you have, because the extra X's are inactivated, so functionally each cell has an X and a Y, which is male. Extra Y chromosomes are also tolerated. This XYY Jacob syndrome, you just end up with a non-disjunction and gives you an extra Y chromosome. At one point in time, this was believed to be the, how did they put it, the serial killer gene. So people that were XYY were supposed to be like psychopathic murderers and stuff, and they found that really doesn't do that at all. There's no correlation between having an extra Y chromosome and that. It was just kind of a theory, well, males are aggressive, so extra Y means more aggressive, so therefore you have to be a serial killer. That's not how that works at all. There are some symptoms with Jacob syndrome, which we'll get into a little bit here. So Turner syndrome, person with a single X chromosome, about 1 in 2,000 live births, so not terribly uncommon. Usually short stature, malformed jaw, web-necked, high palate, tend to have heart and kidney defects. Their ovaries fail to develop properly and do not go through puberty without menstruation, or menstruation without sex hormone replacement. Tend to have some learning disabilities non-verbally. They seem to get lost and forgetful frequently. That's tend to have a fair amount of anxiety and temperamental. But overall, it's not the worst thing in terms of genetic disorders. Klinefelter, so you get a male with 1 Y and 2 X chromosomes, tend to just have a little bit of speech and language delays and may require assistance to father children. Fairly mild overall. In Jacobs, you get that's when you've got the 2 Y chromosomes, so you have a XYY genotype, and they tend to be a little tall, thin, have acne, speech problems, reading delays. Delayed emotional maturity, so they can be a little more aggressive because they have difficulty regulating their emotions. But for the most part, you really wouldn't be able to tell that someone had Jacob's syndrome unless you check their genotype. It's pretty mild. It does not mean you're a serial killer. Poly X females, so triple X or more, they tend to be taller than average. Usually, other than that, usually no real unusual physical features. Generally are able to conceive children just fine. Slight risk for some learning disabilities and difficulties with speech and language skills. Maybe some difficulties with motor skills, and emotional regulation. But overall, they generally tend to fall within the range that is considered normal. Trisomy 21, most common autosomal trisomy in humans, has very distinct facial features, mild to severe mental impairment. The chances of a woman having Down syndrome rapidly increase with age, starting at about 40. And if memory serves, up until about 30-ish years of age, it's only about a 0.5% chance, and then you hit 40 and it doubles to a full percent. But then every year after 40, the probability of having a child with Down syndrome rapidly increases. You can do karyotyping with amniocentesis or amniocentesis or chronic villus sampling. It's basically you can sample the child's DNA in utero, and one of the characteristic physical features is this simian line. Like there's one single line across the palm. If you look at your hand, you've got typically 3, 2 there, and then the one around the thenar eminence here of the thumb. But they just have the one single line, and that's pretty indicative of trisomy 21. And all of that is just summed up right here in this one table in terms of, whether or not you get or what each of these syndrome is, and the gender it affects, or the sex that it affects, the chromosomes, number of chromosomes you end up with, the rate of spontaneous abortion, and live births. For the live births here, it's really talking like how frequently it shows up among those who are born. And the only 2 here that we really don't have good data, poly X doesn't seem to cause a spontaneous abortion, don't have enough data on Jacob's syndrome. So you can also have additional chromosomal mutations when the chromosomes break, either through environmental damages or viruses can cause damage. And typically you repair your DNA by just putting the sequences back together, and it's fine. But you can have deletions, duplications, translocations, and inversions in the process. And this has a lot to do with why women have a much higher chance of having children with birth defects or trisomies or what have you as they age, because women are born with all of the gametes they'll ever have. Technically speaking, you had them before you were born, so your grandmother also carried you in utero for 9 months because you were a single cell inside your mother's ovaries while she was still in her mother's womb. So, interesting. That's something most people don't think about, but it is kind of interesting. There were 2 women that carried you around for 9 months, not just the one. But because of that, all of the things you're exposed to during your life that potentially can damage DNA can potentially damage the DNA in the ovaries, or in the ova in the ovaries, and it's cumulative over time. That's also part of the reason why a fairly high percentage of births result in a spontaneous abortion, often before one even realizes they're pregnant. It's just a particularly heavy or spotty period, that cycle. is all it commonly appears to be. Males, on the other hand, continuously produce new gametes their entire life, which is why a male of 70, 80 years of age can still father perfectly healthy children a very high percentage of the time, assuming that they remain physically capable of doing so, let's say, which is not terribly common. But if they were able to, there's still a very high chance they could have healthy offspring, even late into their life. Which I've heard a theory about, and I do apologize for going off on a bit of a tangent here, but it is interesting. That is, this capacity to father children late into life that promoted the longevity of our species, because normally evolution selects for the capacity to reproduce and bear offspring. And a woman who has gone through menopause no longer has the capacity to have offspring. So they are not being, that extra longevity is not being selected for in the traditional sense. But what happens is, because the males are capable of reproducing so much longer into life, a longer lived active male has a greater chance of passing on his genes to the next generation. And the daughters inherited those genes for longevity as well, which resulted in them surviving past menopause. And that created essentially the grandmother. And having an older, more experienced female around who can coach her daughters and granddaughters on how to take care of and foster children significantly increases the probability of her grandchildren surviving, and therefore her genes are much more likely to be passed multiple generations into the future. And so the two things working two factors working hand in hand ultimately help to increase the lifespan of our species, which is I'm not entirely sure how one would prove that hypothesis, but it makes good sense to me, and I kind of like it. But that aside, that won't be on the test or anything. Just one of my random tangents. Anyway, back to the material at hand. So, changes in chromosome structure. You can have a deletion, which is essentially what it sounds like. A small piece of the DNA is removed, and when it was repaired, that information was lost. And you can have Williams syndrome. As a result of that, you lose a small piece of your 7th chromosome. And I'm not entirely sure I'm pronouncing this right, but the cri de chat, it's French, so I probably butchered that. Any of you who speak French, feel free to correct me in class. It means essentially the cry of the cat, because the children who suffer from this deletion of the end of their 5th chromosome have a very distinct cry, and it sounds like a cat. These are typically tolerated, but there's severe symptoms as a result of it. But that's a whole nother topic. You can have inversions where a piece of the DNA gets rotated 180 degrees, and because you always read the DNA in the same direction, basically everything in there becomes gibberish because you're reading it backwards. So you can result in some strange malformed proteins as a result of that, or odd regulatory factors. It's typically not well tolerated. You can have duplications, and this tends to be the most tolerated of the mutations. Especially, it depends on what you're mutating. If you were duplicating, say, a receptor for taste, then what ends up happening is you can just taste more things, because typically you don't just double a thing. You can have these like duplication inversions, or you can have a duplication and then have further point mutations later on, and it changes the shape of the receptor, and now you can taste more things. Or there's multiple ones for the rods and cones in your eyes, so you can see more colors for individuals like that, which is much more common in females because the ability to see color is on the X chromosome. So if you have a mutation where one of your X chromosomes has a broader range of colors you can see, well, then on it makes good sense that on average the majority of women can actually see a broader range of colors than the average man. If you have this inversion, or this duplicated inversion, though, on the 15th chromosome, then you have delays in motor functions and this characteristic skin fold at the inner corners of the eyes and a flattened nasal bridge. Translocation, you basically just have a piece of one chromosome that gets picked up and moved somewhere else, and typically that's okay within that individual because all the information's still there, and as long as you didn't like pop it down in the middle of like a regulatory system or something, it's probably fine. But you can have what is essentially a functional trisomy within the next generation. I knew someone who had one of these translocations of part of their 21st chromosome, and for them it's fine, but they passed on a additional like one of their other chromosomes had part of the 21st on it, and because it was translocated over from there, like they still had all the same DNA basically, but they passed on this other chromosome that had the extra 21st attached to it, and so their offspring essentially had a functional trisomy 21, even though they only had 46 chromosomes. So it was it's kind of interesting how things like that can affect multiple generations down the line. But that is it for 24. So the main thing is know the types of mutations, know the disorders that can be result from trisomies or monosomies, particularly with the sex-linked ones. Just be able to explain how the non-disjunction in mitosis and meiosis works. You can just look at the different steps on this picture and you're good. If you have any questions or comments, concerns, let me know in class, and I'll see you next time.